It's my pleasure to introduce today's speaker. Dr. Raj Kannan is an assistant professor and senior associate consultant in the Division of Experimental Pathology, Department of Laboratory Medicine and Pathology, with a joint appointment in the Department of Cancer Biology at Mayo Clinic in Rochester, Minnesota. Dr. Kannan heads the Stem Cell and Cancer Biology Laboratory that conducts fundamental research spanning early embryonic to adult stem cell development and directs regenerative biobanks for epithelial tissues, specifically breast, fallopian tube, and salivary glands. He is an expert in emerging stem cell medicine fields with a particular focus on precision cancer prevention and regenerative cancer medicine. Dr. Kannan is an esteemed member of Mayo Clinic Comprehensive Cancer Center and Mayo Center for Regenerative Biotherapeutics. Dr. Kannan's academic journey includes earning his PhD in molecular medicine, where he investigated actionable endocrine therapy resistance mechanisms in breast cancer at the Loggins Institute, University of Auckland in New Zealand, under the supervision of renowned experts Dr. Peter Lobe and Sir Peter Gluckman. He further honed his experience in stem cell and cancer stem cell biology during postdoctoral training at the Terry Fox Laboratory, British Columbia Cancer Research Institute, and the Department of Laboratory Medicine and Pathology at the University of British Columbia in Vancouver, Canada, under the guidance of stem cell pioneer Dom Connie Eves. In his research, Dr. Kannan identified the cellular and mechanistic origins of breast cancers and currently focuses on stem cell biology to comprehend the origin of breast ovarian cancer in high-risk individuals and to develop early interception strategies for cancer-predisposed women. To facilitate this exploration, his team has established unique regenerative tissue repositories from average and high-risk women. Additionally, they have established a salivary biobank to develop therapeutic autologous salivary stem cells for treating xerostomia in head and neck cancer survivors. So thank you so much for joining us today, and I'll turn it over to you, Dr. Kannan.
So my talk is titled An Artificial Intelligence-Powered Approach that Reveals Novel Features of Tumor Cell Metastasis. It's going to be largely basic science, but the way I want you to sort of follow this talk and think about it is this technology enables us to look at cancer from a close quarters in manner that we previously were not able to, and therefore we could probably start asking questions that we never thought was possible, and in the process both reveal fundamental sort of properties of cancer and why is it such a menace and complex disease to deal with, but also along the way discover molecular programs that are very unique to certain complex processes that could then be effectively targeted. I have no disclosures. Today's lecture will help you recognize the importance of tracking tumor development at the clonal resolution. It'll help you to describe heterogeneity in cancer cell growth dynamics, again at the clonal resolution, and also start thinking about classification. How do you want to classify? If you were to study cancer at this resolution, how do you want to now classify cancer cells? Are there new possibilities that emerge from looking at tumor development at the subclonal resolution? And also, what do we learn about metastatic processes, which have been extremely difficult to study? I have a few slides as a way of background, not directly related to clonal investigation, but to really understand how we think about this disease. We all know that cancer is a genetic disease. Most cancers have a very well-described genetic event linked to it. But what is actually emerging, at least in the last decade, is that normal tissues, the deeper you sequence, you actually find cancer-related oncogenic events already present in normal cells. The more you look, the more you find. Almost, it appears like every tissue carries oncogenic events in their normal tissue. and that is normal cells. But it's not that everybody or every tissue has tumors, and therefore we really don't think genetic events are sufficient. They are definitely necessary, but they're not sufficient. And also studies emerging out of Sanger Center in UK continues to baffle us. The mutation rate of different mammals definitely different, but the end of lifespan mutation burden appears to be almost similar across the mammalian kingdom. And this basically suggests that the somatic mutation rate scales across mammals with lifespan. And that would suggest that, of course, the longer you live, the more body mass you gather, and these things would contribute to more mutation in your normal tissues. And there would be oncogenic events also occurring along the way. But that does not mean that you're going to get cancer, or not all those cells would turn into cancer. And therefore, how do we then understand or reconcile this information that you have mutations, cancer-causing mutations in our normal cells everywhere, but we don't get the disease? Or if we get the disease, then how do these actually oncogenes contribute to maintenance or propagation of the disease? So the other sort of piece to this is the developmental biology. And at least since the discovery or the method that was developed in the early 60s by Till and McCulloch, the pioneers of modern stem cell biology, the field as we have recognized today, which is highly quantitative, provides some great insights. Now, the most simplest or most sort of apparent example that I can cite to you is that normal cells, in this case, a pluripotent stem cell, you could get it from the inner cell mass of a blastocyst, the embryonic stem cells, or you could take any adult cell, quote unquote, normal cells, and introduce certain genetic events and then reprogram them to embryonic state. And therefore, they would be considered normal. And since in vitro, they can differentiate into different cell types of our interest, you could further explore them to generate normal tissues, even organs that then could be used for therapeutic purposes. So the very stem cells that can do all of these in a very organized manner, if you inject them in an immunodeficient mice in an adult, just stick them somewhere in the organism, you will see that they form tumor. Normal cells, genetically normal cells, can form tumor. So what does this teach us? It teaches that actually it is the developmental state, or in this case, the primitive developmental state, embryonic or more pluripotent state, that actually has the ability to develop, but when it develops in the wrong place, it appears like it is causing the disease. Now, also from the stem cell field, the other important aspect to remember is that by introducing just 4 genes, Yamanaka was able to reprogram a somatic adult cell like the fibroblast into an embryonic cell. And one of those was actually a proto-oncogene c-MYC. So therefore, oncogenes could turn on programs that are linked to stem cell state. They may not perfectly reprogram them to embryonic stem cell state, but there are now growing evidence that mutations tend to rewire cells to a certain state where they could actually then go on to exhibit some of these developmental properties that we don't usually associate with the site where it is happening. Now, if you read articles on cancer, if you want to like really summarize basically everything that is there in the literature, you will see these points emerging. Cancers evolve through clonal expansion. It's a clonal disease. There is a genetic diversification as the disease sort of evolves. More mutations happen through random process. And so you have this genetic diversity. And then of those genetically diverse clones, some get selected. And that is because they have better ability to adapt to this changing landscape that is dictated by many factors. There is definitely a Darwinian process at play. You have clones that are fittest, that would actually evolve better, survive, adapt better, and therefore have the chance to actually go on and kill the person. But there is also this information that most people, even working in this area, do not remember. And this is very important when you're investigating cancer, that these two types of clonal information could be operating simultaneously. And what are those? When we refer to clone, most cancer biologists today would consider clone being genetically identical, or you could trace the origin to the common genetic event, common genetic ancestry. Therefore, it is really the genetic clone that is being described. But developmentally, that is not how we look at a clone. If you talk to a developmental biologist, it is the common trait that we are interested in. So a cell that you could develop a tissue into, and if they replicate themselves and those cells can then go on to make the identical tissue, then we call that process cloning. It could be, you could extend this definition to tissues, cell types, organs, organisms, essentially a cloning trait that you're interested in. Now, how do, therefore, you study tumor cell heterogeneity? We all know that tumor cells are very heterogeneous in nature, both genetically as well as sort of in their epigenomic states. So how do you sort of look at this heterogeneity? The genetic clone structure would force you to assume that cells actually have different genetic makeup. And when you organize them, you would have a hierarchical structure. It all basically leads to common genetic ancestry. Now, that's what you usually, that is information you actually extract from sequencing any tumor. Whereas in the developmental biology sphere, if you take a tissue or a tumor tissue in this case, you have cells that have different functions. Some have growth ability in vitro, some have growth ability in vivo when you transplant them. Some can form multi-cell type kind of three-dimensional growth. Some only grow as 2D colonies. So there is different types of growth properties that these cells display. So functionally, they can be very different. Also phenotypically, they may express different cell surface markers or biomarkers that you can identify or pull them apart. But also when you start carefully mapping the hierarchical sort of nature of these cells as to who is coming out of whom or who is producing whom, the progenitor-progeny relationship, once you start establishing those through very rigorous experimentation, you will see akin to normal development, another hierarchy emerging that is cancer specific. Now, this hierarchy might look very distorted from what one is used to seeing in normal development, where in normal mammary development, you have the luminal epithelium and the myoepithelium. And you know that there is a bipotent progenitor that actually generates those two lineages. And in blood, you have this common progenitor cells that gives you myeloid and lymphoid. So you have a clear understanding of what you're looking for and how you study those. But in cancer, we don't know what we are looking for because the end product is not well defined. All we know is that there are some cells that have ability to grow and some cells that do not share that ability. And cells that grow have the ability to generate cells that do not grow. And now, depending on the kind of growth assays that you have and kind of nuances in your assays, you could still get a fairly robust hierarchical sort of differentiation model established, and through assays that enable you to see how this structure, this developmental clonal structure, can replicate itself through the process of self-renewal, where the cells that sit at the top of this hierarchy, which we refer to as tumor-initiating cells or cancer stem cells or clone-initiating cells, how do they sort of expand and maintain a pool of cells that that continue to sort of propagate and generate all the cells that form this highly differentiated tumor types. or mixed bag of cells in any given tumor. Now, the cancer stem cell hypothesis also sort of tries to explain how the disease progress to therapeutic resistance. And it may or may not have genetic mutation or structural change in the genome associated with it. Simply by rewiring the cells, you could create a certain state that would allow the tumor to resist therapy. But of course, the genetic event or the constant sort of evolution, you have more options to kind of use the genetic event towards the advantage as in how these primitive cells or cancer stem cells adapt or create more variation in their pool. So a normal stem cell would differentiate. That's a normal hierarchy. One of these cells, let's say, gets mutated and therefore becomes the cancer stem cell that still develops, still is probably responsive to a lot of physiological cues, but it forms distorted clones. And through self-renewal process, the pool of cancer stem cells continue to expand, and it might acquire resistance through various mechanisms that we know of today, and eventually leading to selection of certain clones because of their relative growth sort of advantage or adaptability in the environment. How do we therefore look at, how do we fit metastasis in this whole clonal structure understanding? Of course, you could do the same thing, which is to take DNA from primary tumor, metastatic tumor, look at the genetic makeup and try to map them out. And when you do that, again, very sort of simple structures or these phylogenetic kind of trees emerge where metastatic cells have certain mutations that you can map to the primary tumor, a certain subclone of the primary tumor. And you know that these probably have certain advantage and therefore they have actually evolved to metastasize. But is that what is happening, or can that explain metastasis? Certainly not. And if genetics could explain metastasis, I think we would be closer to therapy for metastatic diseases. But it is a lot more complex. So the cells actually make use of, through rewiring process at the epigenetic level, can make use of the variations that development offers to adapt to the environment and rapidly adapt to environment, and sometimes adapt in ways that normal cells would not. And could be metabolic adaptation or acquisition of an embryonic state, as in the case of pluripotent, the teratoma assay that I was describing. Or it could be a state that would put them off into sort of some dormancy, and then you could wake them up. So there are many things that the cells could actually sort of have as a consequence of these epigenetic rewiring that can take place. The most sort of interesting, to experimental biologists, the most sort of challenging and intriguing observation is that metastasis process, the cells that actually get out of the primary site, that process is very unpredictable. We don't know which cell actually manages to do that. There is no way of predicting that. But more importantly, and the good news is it's a very inefficient process. Not every cell that is in the tumor has this ability. And certainly not every cell that enters into circulation, in the case of using bloodroot for metastasis, not every circulating tumor cell can survive because it's a very hostile environment. So work from Sean Morrison's lab, they very elegantly showed that melanoma, it's a mouse model, in metastatic melanoma cells, when they enter into circulation, they die, provided there is actually these pre-existing clones that have redox pathway altered in a way that they would have more NADPH molecules to counteract any sudden rise in the oxidative stress level in the cells as they enter circulation. So therefore, these cells would survive in circulation and have a chance to colonize a distant site. And by simply modulating the oxidative stress levels or this antioxidant levels in the cells, they were able to wipe out metastasis in the animal model or get very aggressive sort of metastasis, depending on which way the manipulation was. And this certainly tells you that certain clones probably enter, but we don't know which clones enter into circulation, or if there is a subclonal architecture in the primary tumor, it therefore gets very interesting to know, are there specific subclones that have this tendency, and or is it all random? Because in cancer process, or like, sort of when you go back in time, you always hear this argument that these things are random. The cell of origin of cancer is random. A cell gets mutated and you get tumor, but we have debunked that theory. There are very specific cells in every tissues that are actually more vulnerable to cancer initiation. And also perhaps within the tumor, as we learn more about this clonality at the cancer stem cell level, perhaps there are some cancer stem cells that tend to remain benign, and probably there are some clones that actually, through adaptation process, learn to exercise certain molecular programs that would then make them very comfortable to move out and survive in any hostile environment. Can we pull them apart? Can we separate them? Can we quantify these clones? Can we find out in a highly metastatic disease, there are more of these clones, and in a benign disease, there is a lack of such clones? Can we get the molecular signature of such clones that actually have the tendency to enter into metastasis? These are open questions, but actually very important that we understand. And therefore, we start with this assumption that in a given tumor, the tumor initiating cells, or which is also sort of equated to cancer stem cells or clone initiating cells or tumor clone initiating cells. These are sort of used sort of interchangeably, although there are operational sort of nuances to these terminologies, which I won't get into. But for today's lecture, if I say cancer stem cells, they're similar to tumor initiating cells, similar to clone initiating cells in the tumor. But they are rare. They should be rare by definition, and bulk of the cells that do not share this property are produced from these tumor-initiating cells, and therefore we need to know which tumor-initiating cells are capable of metastasizing, and that is what I'll be describing today, what the advances that we have made. And of course, I've talked about the clonal nature, the assumptions that we make as the tumor clone-initiating cells expands through the process of self-renewal in an aggressive disease, or in individuals who have received therapy where most of the dividing cells you have killed, the residual disease probably enriches for cancer stem cells that have adapted themselves better. And that's why when a metastasis, when you see a relapse of the disease, it becomes very difficult to treat, because not only you have woken up the cancer stem cells, you've woken up cancer stem cells that are better adapted and perhaps also have acquired resistance to the earlier treatment during the process of treatment through acquired resistance, or have accumulated mutation over that period, and therefore have additional ways of defending themselves from the next line of treatment. So we need to know about that. So how do we study clones? Sounds very sort of simple thing to do, but experimentally it can be quite challenging. The origins of quantitative transplant research goes back to early 1960s, where Till and McCulloch, this is post-Second World War, researchers very interested to know how bone marrow transplant could save life of lethally irradiated mice through syngeneic transplants. And they accidentally discovered, well, there was an observation that they made which really heralded the discovery of all the concepts that we actually consider cornerstone in modern stem cell biology, the concept of self-renewal, the concept of differentiation. It all came from that 1961 very sort of classic paper, I should say, where well, I won't get into the study. For anyone interested, please go and read the paper. But in that study, they introduced the concept of limit dilution analysis, where you consider, well, I'll explain the methods in detail, but that's one way of detecting clones and the frequency of clone-initiating cells in any given population. The most stringent way of studying clone is doing single-cell transplants, because you won't have any ambiguity that the cell that you injected gave rise to a clone. But then it can be very technically challenging, as one would imagine, to identify the cell that would actually generate a tumor, but also have that cell at such high frequency that you could conduct it in any meaningful manner in a research laboratory where you don't need millions of mice, but a few hundred mice. And of course, the most recent advancement, that is cellular DNA barcoding. Connie Eves, who's my postdoc mentor, was trained by Till and McCulloch in the 60s. And so she pioneered many stem cell areas, including chronic myeloid leukemic stem cells, and also many of the gold standard methods that is used in hematopoietic stem cells, including all the long-term culture-initiating cell assays that hematologists use. We're all from, well, she majorly contributed to those, but her another biggest contribution was clonal investigation, which led to clonal hematopoiesis concept, which today is a lot more attractive to study in the context of various geriatric illnesses and also in cancer research. But also, it is in her lab when I was doing my postdoc that we started playing with this tool, this concept of how do we exploit genetic barcodes or tagging cells with very unique genetic sequences and following them through their sort of developmental journey and to really find out how big a clone that they can form, where do they land up, and how do we sort of map all these clones to get a picture. So some of the reasoning from first principles was through the transmission of knowledge and all these assays that some of them are published and many of them are like we just discuss and debate in the lab, that has really strengthened sort of my resolve that this is probably the best approach to get to this problem. The limiting dilution analysis assumes that you have these rare cancer stem cells in a population of cells, and therefore you could serially dilute your population, transplant in animals when the dilution is such that the cell does not exist anymore in that population, you will have a negative outcome. Until then, every transplant should give you a positive outcome. And of course, you could use a limit dilution analysis based on Poisson statistics. You can actually calculate the frequency of stem cells. There are now calculators available. You just plug in your cell dose that you injected and the outcome, the number of animals that were positive for the outcome in a calculator, and it'll give you the frequency and the confidence interval. The single cell transplants, of course, the biggest challenge is actually enriching for your cell of interest. Since you don't know who the cancer stem cells are, you don't know how to purify them at unit purity. And if your purity is 1 in 10, still it's good. You could probably do some single cell transplants and have few animals in your experiment that turn out to be positive. But if the frequency is 1 in 1,000, you can imagine it becomes a nightmare to get a graduate student who would inject 1,000 single cells and have one animal that may or may not actually develop a disease. More importantly, to load 1,000 single cells in a syringe and walk into that animal facility assuming or hoping that you have actually loaded that single cell and it's actually going to land in that site where you're going to inject, it can be quite traumatic for the student. And it is probably easy to visualize the floating cell in a sort of in a blood system, but if you're working with solid tumors where you have ripped the cells apart from each other using enzymatic and all sorts of mechanical dissociation process, and then hoping at the end of all of that, that you have viable cells that you have somehow purified and then loaded all in the day's job, then go and inject and feel comfortable about it. It can be even more stressful, and that's why this is not done. But there are some papers, and all those papers are from Connie Eves' lab, so I've seen those experiments. I know, like, I'll never do that, or never recommend anybody to do that. And it is that that forced us to think of better ways to deal with this problem. How do you track clones in a population where you really don't know which cell you're tracking? So the cellular DNA barcoding technology, it is NGS, next generation sequencing sort of based, and therefore it helps you do things at scale. But most importantly, you can barcode large number of cells in which a good number of them are your cell of interest, that is the cancer stem cells, inject everybody in a single animal. And you can simultaneously track these competing clones in the same animal and understand their growth dynamics and also all sorts of other dynamics that you're interested in investigating. The most simplistic sort of description of what this technology is this, that you have a plasmid library where you have actually introduced unique barcode sequences in each of those vectors. And this entire library is then converted into a lentiviral library. And so these viruses, each one of them now carry unique DNA barcode, and the diversity, you maintain them at several hundred thousands to several millions of unique barcodes in your library. And then you introduce or infect your cells where a single barcode is introduced in a single cell. So you kind of titrate your virus so that your virus only infects a single cell. And again, you use Poisson statistics for that to titrate your virus. So 1/3 of your cells, if you have injected, you can feel confident that most of your cells are single integration. And then you purify, of course, your barcoded cells, then use them for experiments, whatever that your experiment might be. It could be in vitro, drug treatment, transplants, doing basic science, or any therapy research. But at the end of that experiment, you assume that some clones have expanded. And you isolate the tissue, in this case, probably a tumor. You would isolate the DNA from that tissue, and then use the DNA for barcode analysis. That's the technology that we're talking about. It's actually more easier than it sort of sounds to conduct these experiments. So those who are interested, don't get deterred by the complexity of these slides. It's actually a lot more simple. You just drop the virus on cells. That's how simple it is. So this is sort of the same thing what I just described, but explaining like how it fits in with the cancer cancer studies, that you could have a primary tumor with barcoded cells, or after therapy and relapse, you could have some clones that now expand. Most of the clones probably would die if they're sensitive to the treatment. And then you could isolate either the relapsed tumors or primary tumors or all sorts of metastatic tumors, isolate DNA and just do the barcode analysis. It's as simple as that. And of course, the data you have to now very cleverly analyze and get maximum out of it. The model that we used, of course, has to be a model that we know for sure would work. So we use the ovarian cancer. As you know, it's a highly metastatic disease, very poor outcome. A lot needs to be understood about this disease. The interesting thing is, we call it ovarian cancer, but it's probably not ovarian in origin. In the last at least 15, 20 years, we have known that fallopian tube epithelial cells, the cells lining the fallopian tube, actually are the precursors from which the cells somehow slide through that tube and stick to the ovarian surface and present as an ovarian cancer. High-grade serous ovarian cancers nearly all of them have P53, some distortion in P53 gene. So that's a very good biomarker for if you're looking for early sort of events in the fallopian tube. And the image there shows how normal fallopian epithelial cells that have ciliary cells, secretory cells, and as they acquire P53 signature, your ciliary cells start disappearing. It looks more secretory-like, and then that leads to some early sort of lesions that then sort of start disseminating. And it's a disease that you know for sure you have dissemination, because the way you detect them is ovarian cancer, which is actually a metastatic disease. And so therefore, there is a lot of confusion as when we refer to ovarian cancer as primary, it's not primary. The fallopian should be primary and this should be metastatic. So as an experimentalist, so it becomes very difficult to sort of wrap around the terminologies that is in the literature. But nonetheless, it's a very interesting model. And a colleague, Chuck Leung, and working in Joan Brugge's lab at Harvard, noticed this very interesting phenomena where a mammary cell that actually has the ability to form a 3D organoid structure. These are cell lines, mammary cell lines that retain progenitor activity. So as a single cell, they have the ability to form this 3D organoid structure. And by activating an oncogene in a single cell in that three-dimensional structure, they were able to track that cell that had that oncogenic event and showed that those cells usually slip into this luminal space and start growing as a lesion there. And they call this phenomena translocation. So the cells from the epithelial lining slips into this luminal space and comfortably is able to establish a colony there. And perhaps we are seeing something akin to that, where cells from the fallopian line epithelium is probably just getting out of that space when they acquire oncogenic events and are able to survive the hostile environment of the peritoneal cavity and stick to various surfaces that provides them that adaptation that is required for the disease development. Well, what I'm going to now describe is actually, I'm not going to, we've used barcoding to study normal stem cells in mouse and in human and in breast and all that. But what I'm going to describe is this one study that we recently published where we looked at the ovarian cancer. So it was published as a resource article. We packed quite a lot of information there. So please go and read the paper if you're interested or excited about this approach to conduct cancer research. So what did we do? This was, again, a simplified sort of illustration. We had two cell lines. They are isogenic, which means they're identical, but they differ in expression of one particular gene, and I'll mention what that is. And we barcode them, purify them, mix them equally, and then inject. Where do we inject? We chose two sites. One is in the ovarian tissue, another is in the oviduct, which is the mouse equivalent of a fallopian tube, except it's very hard to sort of inject in that, and I'll mention why. And then you wait for the cells to form tumor, and then, of course, isolate DNA and study the barcodes. The gene that we chose was LRRC15. It's a cell membrane protein. And the work from here at Mayo by colleague Dr. Sridhar discovered this gene when she compared the genetic changes in the debulking surgeries and that of the omental Mets, and found this gene to be upregulated in the Mets for ovarian cancers. And later, I was part of the other study where later her team described the mechanism through which LRRC50 promotes metastasis. So it's a good gene to have in the study to model metastatic events clonally. And of course, this is the later half of what happens after the disease is developed, right? So you isolate DNA, and then you barcode sequence them, and then you put the data through a data analysis pipeline that I'll briefly mention. And this pipeline is freely available in GitHub for anyone to use. And for those who want to know the complexity, this is the slide I didn't want to show to scare off anyone who was actually otherwise interested in pursuing this line of research. There's a lot of multiplexing that happens to kind of make it practical, doable, and also cost effective for research labs to carry out. And but the good news is the computation effortlessly handles the data to unmultiplex, so deconvolute all the data and gives you sort of beautiful results. So it was Dr. Callery, so bioinformatician, and her staff, Xiaojia Tang. They contributed to the development of this clone initiating cell calculator. And the details of the pipeline is, again, simplified illustration of the pipeline is there. And what it does is, of course, it deconvolutes, but also it actually takes care of the error rate, the sequencing error rate, and it also takes care of the thresholding that is required. We also have something called spike-in controls in the experiment. Again, I won't get into the details, but it helps us get absolute counts of these barcodes. So therefore, we could back calculate and actually find out how many cells were actually present in that tumor that is of our interest. So we could actually start quantifying absolute clone size in that given tumor or tissue that we are dealing with. A talented surgeon from Beijing who was doing postdoctoral training at that time actually embarked on this study because we initially did not plan to inject an oviduct. It's a very challenging site in mouse to inject because none of the needles that you have or you could source for me would actually help us inject in that size. So we had to pull the capillary glass in order to get the right size and then use that as a needle to inject the cells in the oviduct. And this is actually a very good model because the take rate is 100%. And there's another postdoc, senior postdoc in the lab now, Mushir Alim, who helped develop the barcode libraries, participated in this study as well. And you can see that injection of these cells by around 7-8 weeks, you get quite lethal disease. You have to sacrifice the mice by then. And I've put the experimental sort of design on the right side so that you can follow the results. So you have the two isogenic lines differing in the single gene LRRC15 expression, barcoded, mixed together equal numbers, injected in ovary or oviduct. Right, so what are the findings? So the first thing is we find that as expected, LRRC15 expressing clones, not talking about cells now, I'm talking about clones, are more fit. You find them to be more fit in terms of their ability to grow in ovarian site when we inject the cells there, or in the oviduct site. And please remember, this is a competition experiment. We have both LRRC negative, 15 negative and 15 positive cells injected, co-injected in the same site. So the clones are trying to compete with each other for resources. So it's actually a good way of comparing their growth dynamics. And also less than 0.05% of cells that we transplanted had the ability to develop into a clone. That's something to, again, keep in mind. Fits in with the theory that only a rare cell can engraft. Even in cancer, not every cell has that ability. And most of the cells, since they don't have the engraftment ability or cloning ability, are not actually, they will actually exhaust themselves if they don't have a primitive cell to replace them. So therefore, it's really important to study these clone-initiating cells. And when you look at the, sorry, this slide has come out very sort of not so clean, but what it shows is metastatic clones and non-metastatic clones of these sort of tumors that developed. And you can see that majority of them are colored. That means they are metastatic, green or red, doesn't matter. So that represents actually metastatic. You can see that most of the clones that were detected were metastatic. So this is actually a highly metastatic disease. To begin with, the cell line was established from a metastatic site. So you would assume that most of the cells still have that features and would behave in that manner. And of course, the other nuances that you see that there's more metastatic percentage of clones in the LRRC15 positive arm of the experiment compared to the LRRC15. But then we also should remember that LRRC15 negative cells are not that fit. So it is among the ones that actually survived, we see this distribution. That's something to keep in mind. Now, what happens when you're looking at all these different sites? Lung, liver, like in whatever vital organs that we were able to isolate DNA and analyze barcodes, we see that LRRC15 positive clones actually outperforming LRRC15 negative clones in all those sites. There is some surprise waiting for you later at the tail end, but that's something to, again, remember. So this basically gives you an opportunity to study clones in this manner, study the effect of a single gene on the clonal fitness. This, again, slide has not come out really well, but we could also take this data and try to fit it in a circus plot that we are used to seeing for genomics. But basically, the outer arc represents various organs from which we have isolated DNA and analyzed barcodes. And now you can start seeing how these complex patterns of dissemination are happening. And the top row is a single animal where you have deconvoluted the data and you're looking at the competing LRRC15 negative and positive clones. And just by looking at that, you can already tell that LRRC15, there's a lot more complex dissemination pattern compared to the LRRC15 negative. And this enables us to now look at clones differently. Now you have certain clones that you only find in the primary. You can see those green lines in the black sort of arc region. That's clones detected only in the primary tumor or at the site of injection. And then you have clones where you actually see them in one other tissue, one other organ. And then clones that you actually can find in multiple organs. And then there are some rare clones that you can find everywhere. Right now you can bin them as different clonal types. And then you can ask, what is the influence of a gene on these different clonal types? And of course, these clonal types represent the aggressiveness of the disease as well. So within a growing tumor, you have all these different clones performing at different rates, and we can quantify them now in any given experiment. So this actually enabled us to look at the distribution, both in the oviduct site and the ovarian. And you see that in ovarian site, you have this TOTI clones appearing, CIC TOTI, that is clone initiating cell with totipotent ability that is found everywhere. So clearly, ovarian site, the clones are a lot more aggressive. But in general, LRRC15 positive cells, those clones are a lot more aggressive because you see a lot more CIC pluri type. So this is one way of looking at the data. And of course, you can take all the barcode data and then scale them using multidimensionality sort of scaling methods. And this is across animals. You see that all the primary tumors actually cluster together based on their clonal behavior and the other organs. And something interesting starts appearing. The organs that are accessed through blood, which is the brain, the salivary gland, the heart, they all start clubbing together. And then the ones that is close to the right ovary where it was injected, which is the right kidney, I don't know what's behind that right kidney, but ascites and also the contralateral ovary, which is all peritoneal accessed organs, they all start clubbing together. And it's also very interesting to note that in oviduct and ovarian, irrespective of the site where we injected or irrespective of animals that were injected, we were able to reproduce these differences very, very well. All the primaries were clustering together in all these animals. So therefore, we started asking this question, are the clonal behavior data, which is quite complex, you have clones everywhere doing various things, can we use this information to somehow start understanding, are there certain clones that have a certain tendencies? The first thing that we did, this is basically coefficient of multivariate linear regression analysis showing clone size that are observed in blood, ascites, and primary, and then ask whether they are able to predict the clones that will appear in the lung, liver, spleen. And you can see that blood is able to predict clones that would appear in the lung, liver, spleen, and all the blood accessed. And then you also see ascites being able to predict left ovary and omental metastasis, right kidney, etc. So that clearly seems like the clonal behavior can be modeled and you could get something out of it. And of course, we did a histology to sort of evaluate that what we are seeing is not an artifact, but real. So these are human-specific Ki-67 antibodies tested on mouse tissues to show human ovarian cancer cells seeding the disease in these distant sites. Right, there was something done to validate that these metastasis events are real. And of course, this forced us to think on a per clone basis. Each clone has a very complex dynamics. It's going to different sites doing many things. Can we use this information to now start understanding the relationship, or there are specific behaviors that we could actually tease out from this data? And what I'm showing here is basically these are Pearson correlation matrix of every clone against every other clone. That information was fed into this, again, multidimensional scaling method or UMAP. And UMAP does a better job where you can see all these clusters appearing, each clone actually falling into one of these clusters. And now you can take those clusters and start actually looking at the metastasis behavior. So this is actually the highest resolution of a clone doing its thing, which is metastasis, but actually you are now able to define these clones based on their common, that is the shared properties. And of course, what would be very interesting to know is the ones that would go on to kill the animal and what is the molecular property of those clones, which is a question that would follow from this research. And of course, we trained the elastic net model since it became quite apparent that the clones, the information was enough to probably tease out the blood route versus the peritoneal route of metastasis. So we trained the model randomly using some clonal data and then tested on the remaining data where we were able to show with very high degree of accuracy and predictability that the model was able to, on a per clone basis, was able to predict the organs that would actually use blood route for metastasis, or the clones that would use blood route for metastasis. And we were able to show that ovarian site, injection in ovarian site, you had more of these clones that would use blood route for metastasis. And this was important because in ovarian cancer research, hematogenic sort of metastasis is not investigated or not considered important because most of the damages is really happening in the peritoneal cavity, becoming quite limiting for the person's survival. And also in the study, I said there was some surprise. So the surprise was we picked 8 bubble mets from the study. These are macromets, probably 1 millimeter each. We should have the scale. I don't know where the scale has disappeared from the slide, but so these are really tiny, but macromets, we isolated DNA and did the barcode analysis. You can see, and since it's a competition experiment, now you can see LRRC15 positive, negative clones in each of these micromets. You can see that 7 out of 8, you don't see any sort of systemically metastatic clone appearing, but 1 clone, which is LRRC15 negative, that appears and seems like it's quite metastatic, that the clone's there. But the clones are there. You can detect them in that site, except they're not actually metastasizing anywhere else. They're only metastatic to the bowel. And of course, the LRRC15 positive, like uniformly, you have these clones also appearing everywhere else or on most other sites. But the surprise was when we actually looked at the clone size distribution of this LRRC15 positive and negative. I showed you that in every other site that we measured, including the primary tumor, LRRC15 negative clones underperformed. Whereas here, in most cases, the clones actually were similarly competent. So there is something about this bowel mets that actually enables, irrespective of which genotype the cell is, it actually promotes clonal growth. And this is important because lethal bowel constriction is one of the problems with these peritoneal cancers. And therefore, we could actually now investigate them. And of course, we have in the process developed an end-to-end clonal investigation sort of pipeline from reagents to experimentation approaches to analytical methods that would allow anybody to conduct such research. And of course, with every research, we'll be advancing how we look at these clones and how we categorize these clones. And of course, using the barcoding technology, we are now asking several other questions in collaboration with various labs, and some of them are kind of listed here. Which ranges from basic science to all the way to thinking about co-clinical trial design or using this to fine-tune therapies for pancreatic cancer, to understanding immunotherapy resistance, and also in breast cancer therapeutic resistance that patients face. And the DNA barcode library that I described is available. But we have also developed an expressed DNA barcode library where you could now combine everything that I just said along with single-cell RNA sequencing technology. You can imagine the resolution that would give you to describe these clones and their molecular signatures. So the library is available. We're working on it, and hopefully there'll be a study that will come out of this, will publish. And also we have actually, we are developing other libraries in the recent years. There have been other advancement for very unique purposes. And we have also, we are in the process of building these libraries, or we have already built the libraries in one of those stages, for these, and they're also available for research. And well, here are the acknowledgements. I want to acknowledge specifically a few people, Mashir Alam and Janning Song, who spearheaded this study, and excellent team of collaborators with Rani Kalari and Shaujia Tang here in the bioinformatics department, helping us with the CIC calculator pipeline, as well as David Knapp from Montreal, who does cell engineering as a day job, but machine learning as sort of a part-time, but really helped us with the ElasticNet model.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.